Okay, this is the exciting part of the day. We're going to be hearing 90-second pitches, right?
Actually, it's up to 3 minutes.
We've increased it to 3 minutes. So good. We're going to hear pitches from many of the entrepreneurs here in the room today. And to moderate and to organize and to run that, let me introduce Ayo Alaran. We actually hadn't. I don't think we'd mentioned Ayo before. But he's very key to this all of the organization of this, and we're just really pleased to have him here. He's not just somebody who runs around with a microphone. He's the man. So yeah, let's give Ayo a hand. But so Ayo will run this session on the pitches.
Awesome. Well, thank you so much, Larry. How is everyone doing? Hope you got some good energy, and we're about to start the pitching activity. So this is going to be the fun part. You've heard it from a lot of speakers. So now is the time for the audience to share your experience, share your companies and your ideas with the judges. So the whole goal of this is we have about 15 or so entrepreneurs that will be presenting tonight. And out of that 15, the judges seated on this front row are going to select 5 entrepreneurs from the 15. And those 5 entrepreneurs would receive coaching 1 hour a month for 3 months from some of our mentors. And some of the mentors are some of the speakers that you've heard from today. So please do your best. You've definitely also gotten some good tips from Chris and from Carolina about how to fundraise, how to definitely share your passion, share your story. The whole idea that part of the reason why we put this particular activity together is for a lot of VCs in the US, they definitely don't have a lot of time. And just think of yourself in an elevator and you have to quickly pitch to or get a VC to be interested in investing in your company or your idea. So this activity is really to continue that process where you look at how do I continue to perfect my sharing my story, sharing my idea, sharing what I'm passionate about. So that way somebody with money who'd be interested, or even not just money, but with the network, who'd be interested in assisting you along your journey. So for today, each pitch is going to be up to 3 minutes. So we have a timer here. So while you stand here by the podium, you could definitely just pick at the timer. Actually, I'll put it right here. So it's right in front of you. So for every pitch, we'll start at 3 minutes. And when your 3 minutes is up, you hear the alarm go off. That means it's time to be done. And then after that, you get 1 minute for questions or from the judges. We're only going to take questions from the judges, just in the interest of time. And before the next person comes up, I'll call the company name and also the representative. So please, as the entrepreneur, please just walk up quickly. So that way we can keep this going in a very good flow. Then some of you have sent us slides. If you've sent us slides, it's on this computer here. I'll help you advance it. Not advance it. You advance it yourself, but I'll help you pull it up when you come up. And if you need to use your own computer, please quickly bring it when I call your name so that way we can keep the flow going. So the very first company that I would like to call is Ikel Labs, Bilal. Is Bilal? Okay, yeah, Bilal, you're first. So let's give Bilal a round of applause. So here's the mic.
Perfect.
You need to present, right?
Yes, that's on the same slide.
Yeah. Okay, great. Thanks, everyone. It's a great honor. So my name is Bilal. I'm the founder of Eke Labs. We empower a user to build the longevity strategy. For this, we propose biological age measurements and also appropriate interventions. So the whole philosophy of Eke Labs is creating end-to-end longevity system. So in the measurements, we have biological age analysis, and also we have some inputs data from different lifestyle applications. And on the treatment, we're developing lifestyle recommendation with some AD partners, and also a bunch of personalized content, and also engaging experiences. So how does it work more precisely? So right now we're on the MVP phase. We released the Glycan test. So this one on the Glycan IgG to measure chronic inflammation and have a reflection on the immune system. We also launched the MVP app, so web app, with stellar recommendation and lifestyle due to this analysis. With the Okinawa Institute of Science and Technology, OIST, we are co-developing a DNA methylation test. And then we have some ideation projects for microbiome, metabolomic pipelines. For those ones, we're looking for partners. So you might have recognized from my accent, but I'm not really from Japan. But we take a lot of pride to build a Japanese startup. So we incorporated in Shibuya. We also have some Japanese VCs backing us up. We are working with Japanese universities. And one of the reasons we are in Japan is that we really want to integrate the pregnancy health system that exists in Japan. I think that's a lot of opportunity for B2B and also B2B2C revenue. And once we're going to launch our inexpensive and highly scalable biological test, we hope to test more with direct-to-consumer testing. And once we have built our brand and once we have built our partnerships, we would like to explore other markets in Asia, in the US, or in Europe by leveraging the positive image that people have of Japan, especially when it's concerning health and lifespan. Whereas, for example, in the US, longevity can be seen as a billionaire thingy. So we try to offer an alternative to this. And yeah, so in terms of teams, so it's myself. So I'm part of the Longevity Biotech Fellowship. Also talking about ecosystem, I was elected as a board member at La French Tech Tokyo. My co-founder has a double degree from Keio University and Central Nantes in biomedical engineering. And our co-founder, CSO, is originally from Colombia, but he worked at Max Planck, at Harvard Medical School. He is also a PhD in the genetic of aging at OIST in Okinawa. And we have several collaborators and advisors in marketing and science. So yeah, that's all for me. Thank you very much.
Yeah, thank you so much, Bilal. Any questions for Bilal from the judges? Questions or feedback?
Great presentation. Thank you. I really appreciate that you're going to be combining multimodal data across a number of different inputs, glycans, methylation, metabolites, microbiome, as well as wearable information. It sounds like EHR records. Would be very curious to understand, and obviously this is a very short presentation, the initial target focus of your go-to-market strategy. What's the smaller customer segment you're aiming to go after?
So at first we wanted to be really direct to consumers here in Japan, but there's just a lot of trust in the public system. So people getting out of pocket for their healthcare is really complicated. That's why we're working mostly with clinics and also fitness studio. So they have a price range between $300 to $700 a month. So adding a longevity aspect to their practice is actually quite interesting for them to differentiate it. And also it's quite easier for them to upsell these kind of products, yeah.
That's great.
You say that you have some VCs backing you up. Who are they?
Yeah, so it's Lifetime Ventures. It's a Japanese VC working with OIST. And yeah, that's our main VC right now.
Okay, thank you.
Thank you so much. So let's move on to the. So next we have Ilichi. Say it right? Yes, Ilichi. Yes, please come up.
Okay, yeah.
Okay.
And how?
Let me, yes, we're
bring
going
it
to, we're going to bring it up.
Yes. Okay. Hello, my name is Yamaguchi, and CEO of Ovidiu Corporation. Ovidiu is established for revolutionize oncotherapy by L-glucose. And what is L-glucose? And L-glucose is an isomer organics of D-glucose, normal sugar, and L-glucose, which is very extremely rare in nature, and normal cells cannot take it. But cancer cells can take it in. And what is the mechanism of it? This is the mechanism. And glucose transporter, each cell has a glucose transporter, and glucose transporter can distinguish the molecular structures. So it can take D-glucose, but exclude L-glucose. However, cancer cells have another channel called voltage-dependent anion channel, VDAC. And VDAC has a diameter with 3 nanometers, and L-glucose can go in the cancers only. And so by using this scientific result, we invented 3 molecules. First one is NBDLG, which shines green after taking in the cancers. And second one is CLG, coumarin L-glucose, which shines blue and also kill cancers because it goes into the nucleus of the cancer cells. And third one is ALG, gold. And gold has is poisonous actually in the cancer cells. So we invented 3 products. And actually, we got animal experiment of CLG. And as shown in this figure, apparently CLG significantly suppressed local progression and distant metastasis of pancreatic cancer. Whereas standard drug, 5-FU, of course, if you got pancreas cancer, you will get the 5-FU, but it's very poisonous and failed to inhibit both actually. And okay, this is our team member. Thank you very much indeed.
Thank you for the presentation. I just wanted to ask one question. Does the L-glucose is being metabolized by cancer cells?
Exactly.
Okay.
Yeah.
So it can be a source of energy for cancer cells.
That's right.
Okay.
Yeah.
Thank you.
That may be right. We don't prove that I think it's almost right.
So in which tumor types would you do your clinical proof of concept study?
Yeah, actually, the first target is the so-called NEN, neuroendocrine neoplasm.
Yep.
It's a part of pancreatic cancers, and also glioblastoma and sarcoma.
And why those tumor indications?
Yeah, actually, it's very difficult to explain, but actually, we got gene panel, and after the gene panel, we could find VDAC appearance from the cancer, each cancers, and finally, pancreatic cancer as the best candidate of appearing as I say, exhibiting the VDAC. And next, glioma, sarcoma, and so on. Yeah.
Thank you.
Thank you very much for the presentation. I'm curious about what would be the original function of the VDAC you presented. What is the original function?
Thank you very much. Actually, VDAC is in the normal cells. VDAC is located in the mitochondria and the membrane of the mitochondria. But after the Wahlberg effect, VDAC is translocated from the mitochondria to the membrane of the cancer cells.
What is the original metabolite for the pore on the mitochondrial membrane? So they didn't require such kind of L-glucose for their metabolism, right?
Actually, we are now investing about it, and it's not yet. I can't say clearly.
What the date you found the evidence?
Evidence?
Yes.
Yeah. Actually, we had actually many experiments actually for 10 years, and 10 years experiment gave this result actually.
Awesome. Yeah, thank you so much.
Thank you.
Thank you. All right, next we have DioServe. Kazuma Kishida. Okay, yes, thank you.
So I'm Kishida. Thank you very much for giving this precious opportunity. And we are Diosive. And yeah, today I'm very excited to introduce our groundbreaking therapy. And actually, these days everyone knows fertility, and infertility is very big problem for many women. And we have very cutting-edge technologies that can convert iPS cells into human eggs. I mean, oocytes. So this is like a mouse experiment, but we induced some genes into iPS cells, and then we converted iPS cells into unfertilized eggs. So, and we tested their viability by fertilizing it with sperm, and then we successfully created embryo. And that started cell division. So that was our invention initially. And that has several characteristics and comparing to the conventional technology. And that is short culture time, and the cost is very cheap. And like even like undergraduate students can reproduce the same process. And actually, the conventional technology required fetal ovaries, but that's very difficult, and we don't need it then. And we can produce eggs unlimitedly. And the technology was published in Nature in 2021. And that is what we are working on as a service. And there are many women who can't have their children and due to their constitutions or age and any other factors. And in these days, people do hormone injection or in vitro fertilization, and just that. Afterwards, there are almost we can say no other treatments, except for using donated eggs. But by using our service, the patient will go to the clinic, and the physician will take their skin sample and send them into like a CPC, cell processing center. And then they will return the oocyte and eggs to the clinic. Yeah, and then they do IVF, in vitro fertilization. So through that process, the women who can't have their children due to their genetic factor or constitution or age and can have their genetic children. So that is very breakthrough. Yes, so this is their team, and I'm the CEO, and actually I was in American Investment Bank before, so my field is finance. But my co-founder, Dr. Hamazaki, who is the inventor of the technology, our core technology, and he is currently in University of Washington as an assistant professor, and he decided every research strategy for us. Yes, and yeah, we have a very strong team and from global institutions. And yeah, our vision is providing novel possibilities for all lives. I can see that. Oh, yeah.
On time. Go ahead, Shane.
Thank you for the presentation. I have one question about the regulatory path. Have you talked with FDA or the PMDAs, and what are the guidance from them? And second question relevant to that. But how do you think about the safety issues for growth to path to the growth?
Thank you very much for your question. And for the first question, regulation. Actually, I talked with a British authority. And I don't think we can do the business in Japan or in the States because in Japan, of course, there actually a regulation exists to prohibit this technology. So we can do this in Japan. And in the States, the situation is very complex, including abortion. So I don't think that is a proper market. So I try to focus on a British market. And actually, they have a good foundation to understand their evaluation, how they can evaluate that safety and benefit. So yeah, that is actually the confidential part, but yeah, we proceed with the discussion. And when it comes to the safety, of course, we do animal experiments, including mice and monkeys. And yeah, that is very critical part. But actually, we include like a human embryo study at the same time. And we currently have 14-day rule, so we can't grow an embryo outside of the body over 14 days. But that has been like there are possibilities that can change. So then we can evaluate the like quality of embryo for longer time. And yeah, by accumulating that kind of data, we can show the safety. Yeah.
So I actually had a pretty similar question, but you're doing this in an autologous manner, right? Is it
Sorry again?
autologous?
Also,
Autologous? Jikateki?
also, yeah, yes,
Yeah,
yes,
it's
sure, sure.
autologous, right? So you're gonna have to do QA/QC and release
Sure.
per patient. So can you really keep the cost down then?
Yeah, the good point for this technology is that we don't need to like duplicate the cells for like 100 million cells or not. We just require a few cells. So like the process of like inducing the oocytes should be much smaller than the conventional other like iPSC derived products.
I see. I guess my point was, depending on your regulatory requirements, right? QA/QC release per
that's
patient
right.
on a per patient basis. That's what's really going to determine your COGS, I guess
right.
was my point. But thank you.
Any other questions? Oh, Carolina, please go ahead.
Of course, we will expand our business on global market. Yeah, initially we go with the UK, but of course the US is very big market and the Japan also.
All right, awesome. Thank you so much, Kazu. So next we have another Kazuyoshi Miyawaki with Physiologus Technologies.
So I'm Kazu Miyawaki, the managing director of Fujiyoshi Technologies. We are not biotech company, we are a medical device company. So what we are producing, what we are developing is hemodialysis device, which is dedicated for home usage, so home hemodialysis. So as you may know, that chronic kidney disease is one of the biggest health problem, not only in the US, in Japan, in Taiwan, in China, in Europe, almost all over the world. In US, there are 550,000 patients taking hemodialysis at clinic. In Japan, there are 340. In China, over 1 million now. So imagine that if we have to take hemodialysis at clinic 3 times a week, one treatment takes 4 hours. So which means that 3 days of a week are dead for you. Can you work? Can you make your life happy? So that's the reason why we are developing this kind of device. You can take hemodialysis treatment at home in a living room. You can talk with your family. You can have, maybe you can work remotely. So it changes drastically a life of a patient. As you may know, that, in-center hemodialysis on the left side. So there are a lot of beds. In Japan, it's in the bed, on the bed. But in the US, you have to take a sheet. You cannot lie on the bed. So it's very, stressing treatment. So every 3 times a week, 4 hours of treatment. And in Japan, this is the situation of home hemodialysis. So I said that there are 340,000 patients in Japan, but only 700 patients are taking home hemodialysis. Only 700. In the US, there are 10,000, still 10,000. But the number is very small because of the device. You see that it's very complex. You have to pipe everything because it's a professional usage. So patients have to take training to use this device. And plus the safety problem is here because this device is for hospital, the clinical workers are besides. But when you take a treatment at home, there are no clinical workers. That's the reason why we're developing this type of small device with safe treatment. What does it mean, safe treatment? It can see the condition of patients. This is a world-first technology introduced into this type of device. So now we are currently fundraising for next series for developing, finalizing the prototyping plus clinical, non-clinical tests. So today I'm here to see some, investors to say hello. Thanks a lot.
Yeah, thank you. Any questions from the judges?
I really like the idea, actually. Maybe two really quick questions. One, the impact of GLP-1s, reducing obesity and the impact on the market of dialysis is the first question. And then the second is, what's the clinical development path or something like this? Do you need to do a non-inferiority study versus patients receiving dialysis in centers? How do you do the study?
So first point about first point is,
GLP-1s, GLP-1s. GLP-1 agonists reducing obesity and the impact on the market for dialysis.
for the market for the dialysis in a clinical point of view, the business, let's say business of the clinics, it doesn't change a lot. It gives a higher profitability because they take the treatment at home, which means that they don't need any fixed assets at the clinic. And for the entire medical, yeah.
So that's why, data,
it gives of course it gives negative impact. So in the future, what our plan for the exit is that M&A for these kind of big companies, big Fresenius or this kind of thing. So and the second point is about study. Is we have to do that on the clinical trial as well at the clinics. Yes.
But you need to compare yourself against patients receiving dialysis in the center.
No, what we do is that only safety, because the dialysis is taking some kind of get rid of the toxins, the water, that kind of thing. So we made sure that it works, and then we put it in the clinics for the clinical trials to see the safety only. This is what we are discussing with the PMDA right now.
May I ask about the technology background, how to minimize the bugs you showed to us, but yeah, I couldn't find out.
Yes, one of the biggest points is that we don't use water. Currently, the water, RO water is required, but our system does not require water. So this is one of the another technology which we install inside this machine.
Are there any concern or you need to confirm the risks of this technology to minimize the aggregation of the blood or coagulation of the blood or something like that?
Because regarding that point, we don't change the technology for the treatment itself. We change the supply of dialysis. That's the point. That's how we make it smaller.
Okay, awesome. Yeah, thank you so much. Oh, we have one more question. Yes, please go ahead.
But I'm just wondering how much it cost for the one
device?
implement. No, for one implement.
One implement. You mean one
So
treatment or
to the cleaning of the
one
blood.
machine?
Yeah.
One machine.
No. So one implement for the one patient.
One patient. You
Yeah.
mean one treatment?
Yeah. Yes.
We estimate that we don't change a lot compared to the current system.
Mhm.
We haven't fixed it. We have to talk about with the agencies in the future.
So in my understanding, is a conventional machine required a lot of water, right?
Yep.
And that costs a lot.
Mhm.
So that compared to the current conventional machine, so that
We don't use water, but we need some kind of the cartridge which recycles dialysis.
Yeah.
It
So-
costs.
okay. So that cartridge, the water, the lot of water, so which is more expensive for the
Cartridge.
patient? Cartridge.
Cartridge,
Okay. That is a disadvantage, right?
Thank you so much. Oh, and to our judges, please, when you ask the questions, please, let's use the mic so that way the interpreters could pick up your questions. Thank you. Next, we have Kenichiro Kuninobi. Yes. Yes.
Okay, so my name is Nobu from Helix from Fukuoka, Japan. So, but now we have a US footprint in J Labs, New York City. So I will share my story. So we started last year with our co-founder, with Dr. Kenichi Yamada, and we moved toward, I have personally more than 15 years experience in the pharma industry. Then from January to December, we expanded our team. So very seasoned, the investment, clinical trial development, CMC, regulatory. So this is our core team at this point, at this day. And also we gathered a seasoned advisor in Japan for the preclinical to the medical in the US, and a bunch of experience in the clinical trial development with also the IP expert. So using those members, so what do we do? So we, in the first target, but we have two pipelines for the therapeutics focus for the ophthalmology. The first one, we focus on retinopathy prematurity. So this is a leading cause of the childhood blindness. So this is, we plan to target the US market by the drug repurposing. Since the neonates, our target is neonates, that's why safety is very important. So we use drug repurposing. And we submitted the orphan drug designation this month, and we will have some certain response next year. And next one we target is the AMD. This is a leading cause for the all generation of the blindness. So, so currently this is a more than, I think, 10 billion or in the future 20 billion market. So we use our new chemical entity to a little bit earlier stage than current standard of care. So those two pipelines, so we raised a seed funding last year, $2.5 million US, and we have some milestone committed to the existing investors. And we finished pre-IND meeting with US FDA this June. And also we finished CMC formulation for the clinical trial batch manufacturing. So now we are targeting to do the IND next year, first quarter 2024. Then, so now we need money to run a clinical trial. So as I mentioned before. So using this money, we plan to have the top line data of the phase 1-2 clinical trial in the US. And also we moved our preclinical asset for the AMD to IND, we hope. So we have 3 investors in Japan for the seed funding. They committed, but they do not lead. So we need lead investor. And we expect to have some value inflection in 2024, 2026, no, 2026. So this is a good timing for the exit. So that's it. Yes, thank you for your listening.
All right, thank you. Well, Carolina, please go ahead. You have a mic is right here in front of you.
For the first asset that is a repurposing,
Yeah.
right? The repositioning.
Yep.
Is the IP
only the
expire, or what's the situation with the IP?
The IP of the composition of the matter expire. But
Okay.
we have a method of use patents granted in Japan. We are still working in the US.
Okay.
And we hope to have some formulation patent.
As well.
Yeah,
Okay.
we try to work.
Okay, thank you.
I've got a question. Could you tell us a little bit more about MOA, how your drug works?
I forgot to mention about. So yeah, so the co-founder, the Dr. Kenichi Yamada, yeah, since this is 3 minutes, so I emphasize our story. But he researched about lipids, especially the oxidized lipids. So this is a target, and those patients, the preterm baby are exposed to the high oxygen. So we reduce the stress by the small molecule with a very safety.
Radical scavenger?
Yeah, kind of, yes.
Okay. Thank you.
Final question. I would like to know about, how do you select ROP as a gate indication? Because, when I think about the exit strategy, so maybe it's a premature segment is very tough for the pharmaceutical companies. So, but some kind of balance is needed, but you are needed. I'd like to know about your thought opinion.
Yeah, this is very rare disease and very important, but Well, as far as what we surveyed, since this indication was already developed by the Takayuki and Shire, so they projected 200 to 300 million annual sales for this indication, exactly same the administration patterns. So we put this top line, the revenue top line.
Any other questions? Okay, one more from Shin.
Can you tell us about the history of your compound and what it was developed for and any talks related?
Yeah. So since this is a drug repurposing, I do not disclose the exact molecule, but yeah, this compounds is some long history of safety and also some exposure for the neonates in the past. So some preliminary PK data we already have in the neonates. So yeah, those safety aspects we summarize and share to the FDA.
Awesome, thank you so much. So next we have Ken Tajiri with X-Forest Therapeutics. Yes, please go ahead.
Hi, my name is Tentajiri. I'm a CSO of Express Therapeutics, and this is Georg, my colleague and director and head of R&D. And we are Kyoto-based startups originated from Kyoto University IPSR Research Institute. And our business is to provide drug discovery platform for RNA targeting small molecule to pharma companies. So let me talk about the background and the key issues of this field. So there is already a RNA sequence targeted drug such as antisense oligo, as known as ASO. And this approach is pretty good, but ASO still has a difficulty in drug delivery. It usually accumulates to liver, and high production cost is also the problem too. And when the modality is small molecules, the low affinity and low specificity is the problem. Therefore, RNA structure is attracting attention these days. It's known that RNA has a keyhole structure like protein has. So one of the good examples for this field is risdiplam, as known as Evrysdi. And this is a drug for SMA developed by Roche and PTC Therapeutics. And the sales of this exceeds 1,000 million euros in 2022. But this drug was founded in phenotypic assay systems, which means this drug was founded from RNA-oriented screening systems. So still a systematic screening system is needed. So here I'm showing the advantages of our technology. So we have a patented RNA motif designing technology, which enables us to do the massively parallel library versus library screening, over 1,000 of RNA motifs and over 100,000 of compounds. And from the screening, not only the intensity of the hit pairs, but also the selectivity of the hit pairs can be acquired. And since the assay was conducted in the single well, so the data quality is quite uniform, which can be a good data source for machine learning. And here is the track record since our foundations. So some of the top Japanese pharma already do the collaboration with us, including Takeda, Daiichi Sankyo, and Kyowa Kirin, and Otsuka. And all of the project has upfront and milestones and royalties. And through these collaborations and through the internal research, the vast amount of data are acquired. So from this year, we initiated the drug discovery program. I'm not going to go into it, but the key strategy behind it is to use our acquired big data for machine learning predictions in order to reduce the R&D costs in the upstream phases and to collaborate with the pharma companies with strong capabilities in the downstream phases to make our business more profitable. That's it.
Right, so
thank you very much.
Thank you.
So can you tell us a little bit about your deals that you already signed up with? I mean, whatever you can share with us.
So,
The deals that you, I mean, you say that those companies, Takeda, and you already have deals with them.
yes.
Can you share a little bit of the something?
Yeah, it's a very long story, but yeah. So first, we make a contract with Takeda. That was 3 months after the foundation of the company. So yeah, so in this in this case, so we had to negotiate the sharing the information of the compound structure and the result of the experiment, and also the royalty milestone, so on and so on. And that was the most toughest part, I think.
As a quick question, because I love this and have actually invested in something similar. Are you thinking about, especially now that you have these partnerships as you generate these data, over time doing your own internal pipeline of for developing your own therapeutics as well?
So you mean the collaboration research or internal research?
Internal research.
Internal research. Yeah. Yes. Maybe in the future we may find the hit compounds, a lot of the hit compounds, and for some compounds, we will do the collaboration with pharma companies. And for another potential good hit compounds, maybe we will make pipelines as well. Thank you.
Yeah, could you tell us the most advanced stage of development of compound, you discovered under your research collaboration or as an in-house program?
Yep. Of course, I cannot talk detail, but one of the compound has a cellular activity already.
So, Arrakis and Ribometrics and other companies have been trying to do this for several years, right? Spending hundreds of millions. Sounds like you have a cellular, functionally active compound.
Yes.
What do you know about the selectivity? Because one of the challenges is that RNA structure is pretty floppy.
Sorry, stability?
Selectivity.
Selectivity.
The RNA structures can be relatively floppy.
Yeah,
right?
yeah. And we define hit compound as the intensity and selectivity at the same time. So of course we have a selectivity. Of course the compound has a selectivity. Sorry.
It must be a simple question, and it must be a little bit challenging, but you had a lot of collaboration with the pharma companies already. And if your technology is will be the special one that they will acquire the company, so more earlier time, but that they didn't so far. What is your explanation for that?
So you mean some company offering to buy us? What did you say?
No, your company, many pharmaceutical company is already looking at into your technology.
Yes.
And if your technology is a good one, the person should be deciding to acquire your company. But not yet. How do you explain the story of your history of your company?
Maybe, it's very cutting edge of the science. I mean, the targeting RNA itself is the challenging area. So maybe that's why the company, some company doesn't decide to acquire it. Yeah, and we also did an investment from R&D and Takeda. That's why, maybe.
You have investment from Takeda as well?
Yeah, that's very small amount, but yes.
Okay. But I think because the assets are not yet in the clinic, they will not acquire them till the assets have proven clinical validity.
How far are you from
Well, maybe it takes another 3 to 5 years. I don't know, but. Drug candidate. Yeah.
Awesome. Thank you so much. So next we have Koji Tanabe from IPEACE.
Hey, yeah, my name is Koji Tanabe. I'm a founder and the CEO at IPS Inc. So the process of development, all your body cells is originated from the fertilized egg. That we can reprogram your cells to the younger state. So that one of the Nobel-winning technology of the iPSCs enable what it can be do this. So similar to the fertilized egg, iPSC can become any type of the body cells, and they have infinite capability for expansion, enabling of the leveraging these capability of the iPSCs enable the next generation of the cell therapy, drug discovery, rejuvenation using your own age-reverted stem cells. However, the mass production of age-reverted stem cell, the iPSC, was a formidable challenge. So the historically limited to the 1 person per year. However, we overcome the obstacles by implementing the mass production system and automated factories. So these our innovation goes beyond the iPSC manufacturing. So I think we can mass produce the couple thousand people's clinical grade cells product using our automated system. So this revolutionized the manufacturing of the clinical grade cell product and to open the door to the next future of the cell therapy and drug discoveries. So I think we are providing our clinical cell product over the 20 global pharmaceutical companies and also global startups. And as a CDMO business, and last year we started to produce the personalized iPSC banking service in Japan. We opened a clinic in the Peninsula Hotel in Tokyo, and we are, I think, leveraging these mass production capability to change the world. So we are envisioning the future where I think everyone can use cell product from the birth, and we will become a global platform pharmaceutical company in the era of using cell product as a medicine. And now I think we are fundraising $30 million to accelerate our business. Thank you.
Amazing technology. Did you use this technology to produce other cells than iPS?
No idea. So we succeeded to make iPSC now, and we are developing developing the automation system to manufacture the iPSC derived cell product as well.
Thanks so much for the explanations. I'm curious about, what is the future status of the cell therapies as a market, market exists. I suppose they're currently a little bit tough situation by now. So I'm very curious how your great technology was spread in the world.
Yeah, because first bottleneck is a mass production, right? I think Japan invest $120 million, I think for 5 years, but they make only 4 people's iPSCs. I think we should mass produce. And the second, the cost, right? Because they spend $20 million, but we succeeded to cost down the $20 million to the $30K to produce the clinical grade cell iPSCs. And the next question is, I think Mori-san asked, I think how to mass produce the iPSC derived cell product. Probably I think we can, right, cutting the edge, that we can change these situations using this technology.
Got it. So you think mass production is one and only the bottleneck of the cell therapy?
Not only one. The second is how to do the, I think, clinical application, right? Because if we do the cardiomyocyte seed, how many people we can make a surgery per day? Because if we can do 2 people, how can monetize in the hospital? That's also next questions. That's why I think a cardiomyocyte, I don't know. But immunocell therapy, probably I think feasible because we can inject a lot of people, the immunocell to a lot of people in the monetization. We should think about these things. And also ecosystem, right? Because just we cannot, I think, we should not, I think, cost down the 20K to the 100 yen. It's not, right? Because I think we should make an insurance system to realize this ecosystem in the global, yeah.
Last question. And have you communicated with CDMO company like Lonzers and CDI or something like that?
That's a competitors. We are competing with them, but I think GMP manufacturing, probably I think our share is higher than them. Yeah.
For your presentation, I wanted to understand your core part of technology that enable you to do the mass production. Why your technology enables to do the mass production?
Yeah, if you can simply automate the conventional protocols of an iPSC, it's impossible. You can automate, but you cannot minimize minimize the automation system. In that case, we cannot put the lot of system in the loom. We can make, I think, one single automation system in the loop. So that's the most important things. First, we almost reinvent the process of the iPSC reprogramming process, and then we like the lock button, and we automate it in the simplify in the system.
So you have to make, you have to reinvent the, process, product by product basis, or you have some standardized method that can be applicable for multiple.
I think so. I
products.
think so. For example, right, because the people use a centrifuge to isolate the PBMC from the whole blood, but I think we should isolate the PBMC, right? But I think we succeeded to isolate the PBMC without using a centrifuge that can use for the CAR T cell therapy as well. So that I think that's why I think we can. I said I think we can leverage this technology not only for the iPSC manufacturing but also for other cell therapy as well.
It's very interesting. So you said you're already making GMP grade?
Yes, I think we already provide, and one of the couple partner is I think going to CDE and also FDA, and a couple I think 4 or 5 partner is going there. We got the already.
I see. So you're karyotyping and then post, iPS production, differentiation into other cells.
Yeah.
Can you automate that part as well?
Yes, the karyotyping is not enough now. It's FDA is asking us the cancer panel with the next generation sequencing, and also I think sterility test. I think that's deep enough in the USP.
Oh, okay, got it.
We have a QC lab in California that's it's accredited by the ISO.
Okay, got it. Thanks.
It must be very difficult to get customers because as a CDMO, you want your baby to be manufactured in a company that you already know, like the Lonzas, Wuxis, and so on. How are you planning on growing so much?
Yeah, because about this market, because because after getting I think approval of the I think some iPS derived cardiomyocyte, WuXi should do that because they should make a 1 × 10⁻12 cells or 19 cells, and that's their part. But we are focusing on to make a seed stock of the iPSC from the multiple donors at the same time. So that's our strong capability. And first, I think then they're expanding the cell product. That's, I think, a WuXi or Lonza's, I think, task. That's not us. So that's, I think, our standing point in this field.
Thank you very much for interesting presentation. And I believe that this machine would contribute to the quality control instead of the cost reduction. But I'm also curious about how much this system reduces the cost. Do you have any comparison?
Basically, a simple question is, I think Japan spent $120 million for the 5 years, and they make 4 donors. That's the indirect cost. It's a lot, right? But even the direct cost, they spend $20 million per person. But I think we are providing $30K to make a personalized clinical grade iPSC now, and we are already providing service for them. That's not, I think we cannot, I think, shorten the time period, but we can mass produce the couple of people's iPS cell at the same time in the same room because this is closed system.
Awesome. Yeah, thank you so much.
Yeah, thank you.
So next we have Naoto Fujio with MA.
Okay. Hi, my name is Naoto Fuji. I'm
Just a sec.
Thank you. So, my name is Naoto Fuji. I'm CEO of Biofinoma. We have core technology called TN Cyclone ultra-sensitive ELISA with GeoNAT. So please remind the COVID-19 pandemic. So we used the PCR test as a diagnostic test, golden standard. However, we cannot detect the timing when we get over it because protein is not detected. So yeah, key point is to analyze biological phenomena mechanism is protein. So protein is needed. So we decided to found the company with Professor Ito. This is a team. So this is also our mission and vision. So for example, in the case of the Alzheimer's disease, so highly invasive diagnostic is needed. Probably in the near future, blood test would be applicable. But we aim to the more good future vision to create. So our mission is to create an innovative platform that allows anyone to easily detect ultra trace proteins. Our service, our technology has three characteristics. The first one is the ultra sensitivity. The second is a simple process. Third is a simple device. So more affordable and more easily. So the technology is produced by combining the two conventional assays, sandwich ELISA and enzyme cycling assay. To optimize for reaction, we created a unique substrate. Third point is by choosing the appropriate second antibody, we can quantify a variety of proteins. So our primary customer is academic professor and pharmaceutical researcher who get interested in our technology. So we are working on the clinical data. We are working on the established clinical data. Thank you very much.
Thank you. Any questions from the judges? Yes.
For your presentation. So I wanted to understand applicability of your technology for variety of proteins, So can you apply this technology basically for all the plasma, proteins?
Yes.
Theoretically.
Basically, we can applicable. So this is a second antibody. This is so unique. So we by choosing the appropriate antibody, we can detect, quantify protein.
So could you share, do you have some examples of biomarkers that can be measured with your technology? Or that, because of your technology, is there any PD marker that was enabled to be measured, I'm very curious because, your technology is very sensitive and, very low concentration, maybe enough to be measured, right? So maybe because of that, your technology may have enabled measurement of some really difficult to measure markers, and I want to understand examples for such kind of things.
Thank you very much, your question. So so amyloid beta is taking attention from the market. It is very difficult to detect. But we have successfully detected amyloid beta in blood. Yeah, that's one sample. The second point is GRP78 contains the exosome promotes the stress cancer cell. We can also quantify this element.
Measure beta amyloid in patient plasma samples. Wow,
yeah.
That's interesting. Have you collaborating with some academic people so that you can accumulate data of the beta amyloid levels in Alzheimer's disease patients?
Yeah, we have studied the discussion with some academia.
Okay, very interesting. Thank you.
Yeah. Okay, awesome. Thank you so much. So our next presenter is Shinji Kagawa. Actually, Shinji will be presenting in Japanese, so we're going to pass this set to our judges. So please switch to channel 2 to listen to Shinji. So Japanese. So any member of the audience, if you need to listen to this in English, please switch to channel 2 on your interpreting set because he's going to be speaking in Japanese. Please go ahead.
Thank you for the opportunity of this speech. I am Shinji Kawaya from S&K Biopharma. それではここから日本語でなさせていただきます。弊社はですね、安全で優しい医薬品、特に薬効の高いものを患者さんに届けたいということでですね、多機能性タンパク質のラクトフェインに着目して医薬品の開発を行っております。通常、ラクトフェイン、血中安定性が低いため、安定化させるようなタンパク質を融合させた2種類のシーズを開発し、それぞれ分子標的薬として研究を進めております。まず、ラクトフェインですけれども、鉄を結合するタンパク質で、牛乳よりも母乳、特に初乳に多く含まれております。免疫調整作用とか、抗炎症作用、高腫瘍など多機能を示すということからですね、2番で示しているような疾患について治療効果が報告されております。それでは弊社シーズについて簡単にご説明します。以前開発されたものと比べて我々の方は腸細胞を用いているため、静脈や局所投与が可能。また IgG の FC 領域やヒト血性アルブミンを融合させることで、血中安定性、半減期がですね、大幅に向上してですね、薬効の増強も確認しております。それ以外に FC 融合型はですね、分子標的として好中球細胞外トラップ、ネッツと HSA 融合型はコンドロイチン硫酸塩を分子標的として医薬品の開発を進めております。1つ目のシーズ FC について簡単にご説明しますと、まずですね、好中球にネッツを誘導させると緑色のように染まりますが、そこにラクトフェインを添加するとネッツの減少、FC 融合型にするとですね、さらなる減少と正常細胞の増加が認められました。ネッツを形成する背景症モデルを作成してですね、微静脈投与を行うと、発症の遅延と生存率の改善が認められております。もう一つのシーズ HSA 融合型はですね、コンドロイチン硫酸塩の阻害剤として開発をしております。神経細胞にコンドロイチン硫酸塩を添加するとですね、軸索の伸長が、阻害されますが、そこにラクトフェインを添加すると正常細胞程度、HSA 融合型を添加するとさらなる伸びていることが分かりました。そこで、片側、右側ですね、脊髄損傷モデルラットを作成して、髄腔内に、連続投与を行っております。28日目の動画を見ていただいて分かりますように、HSA 融合型はですね、正常歩行に近づいております。現在のパイプラインです。脊髄損傷、肺血症、他に指定難病を中心にですね、動物モデルの POC を行っております。市場規模としましては、4疾患すべてについてですね、2割程度が投与していただければ63億ドルの市場を獲得できるつもりでおります。これが現在のチームです。ご清聴ありがとうございました。
Thank you. Any questions from judges, please?
一点お伺いさせてください。鉄の代謝とミトコンドリアの障害っていうのが結構私個人的に興味があるんですけども、ミトコンドリアの障害がある状態で、このラクトフェリンがですね、その障害をこう直したりとかですね、ミトファジーを活性化したりとか、そういうようなデータってお持ちじゃないでしょうか。
ですね、以前所属してたところでですね、肝炎でやってまして、ミトコンドリアとか、フェントン反応を抑制することは分かっております。そのためミトコンドリアの、DNA の、ダメージを抑えることは分かっております。
ありがとうございます。なんか結構ミトコンドリア障害が鉄の放出がいっぱい起こってですね、一見鉄不足みたいになるみたいなんですけど、実は鉄が余ってるみたいな状態があるらしくて、そこにこうラクトフェリンってどう関わってるのかなってちょっと知りたいなと思いました。ありがとうございます。
ありがとうございます。ちょっとメカニズムが分からなかったんですけど、鉄吸収が MOA っていう理解でよろしいんですか？
ですね、これは違います。モード・オブ・アクション、ラクトフェリンって多機能で、受容体が疾患によって違って、いろんなメカニズムが変わるようになるんですけれども、そのために今まで薬になりにくかったということで、我々それを打破するために、HSA 融合型はコンドロイチン硫酸インを阻害する。コンドロイチン硫酸インの受容体に結合して、中和するという感じですね。で、あともう一つが、高中級の細胞外トラップって、熱って COVID-19 にもよくの増悪にも関係するような因子なんですけど、それを抑えるというのが MOA として挙げております。
ありがとうございます。MOA 自体はこうやってバリデートされてるっていう理解正しいんです。MOA 自体はターゲットはもう ID 確実にされているっていう理解でよろしいんですよね。うん。もう一個聞こうと思ったんですが忘れちゃったんで、また後ほど。はい。出しました。これ、アニマルデータっていうのは予防等ですか、それとも治療効果ですか？
ですね、肺血症は手術してから、後に打ってます。で、脊髄損傷は同時投与にしておりますけれども、今後ちょっと7日目以降とかの投与もやってみようかなと思ってます。
多分基本的には予防は無理だと思うので、治療効果、どのぐらいのシビアリティから入れるのかっていうところとかは、すごく気になりました。
Any other questions, comments? All right, thank you. So next we have Takeo Tanaka from Medigay International. Oh, okay, he's going to speak in Japanese as well, sir.
アカデミア発のスタートアップですが、正確に言えば B スタートアップです。B スタートアップですね。僕は目指しているのは、新しい、セラピーなんですが、主にレーターステージのがんの患者さんですね。いわゆる緩和ケアのステージに移ろうという患者さんをターゲットにしています。したがって、薬による副作用とかですね、レジスタンス、これを避けようと。体に負荷があまり大きくないようなセラピーを考えました。使うのはですね、ここに出ていますけれども、非常に小さな微粒子ですね。バイオディグレーダブルな生分解性の微粒子です。これをですね、主要組織のところにシールドをして酸素と栄養を遮断するというメカニズムとしては非常に単純なものです。これは動物実験の結果なんですが、先ほども前のセッションでありましたけれども、まだまだアカデミアでのですね、データギャザリングで、新しい会社ではこの辺をプロフェッショナルなデータを集めたいと思っています。一応このデバイスでもって、主要部分を囲むことができると、完全に壊死に導くことができるかな。酸素を遮断することと栄養を遮断すること。これ一応確認できていますので。先ほど申し上げましたように、ターゲットにしているのは、この、いわゆるパリアティブケアに移行する患者さんたちですね。約世界中で50万人いますけれども、そのうちの数%が取れればいいかなと考えています。これをやろうとしている今、チームですけれども、 まだ会社を起こしてませんので、すべてボランティアで協力いただいています。アメリカにも同じようなチームを作る必要があるかなと考えています。はい、以上です。
Thank you. Any questions, please? Oh, yes, please, let's get the mic.
Okay. 今回の緩和ケアの患者さんが対象だということなんですけど、
はい
すみません。治療の目的は何になりますでしょうか。
一つはですね、今、オピオイド系の鎮痛剤を投与しますね。このステージの方たちっていうのは治療を行っていないんですね。したがって、その痛みを除去するには2つのアプローチがあって、痛みの原因の1つが、腫瘍部分の血管も多いこと。もう1つは腫瘍自体が増殖していることですね。この2つを解決しようということで、1つはこれで新生血管をなくしてしまう。もう1つはそれによって神経を圧迫している圧力を減らすという。それから腫瘍の増殖を抑制する。この2つが目的です。
何か骨髄等にですね、こう入り込んで、私の親会社も一応こうフェンタニール等をですね、販売しているので、こう痛みに関してもなかなかですね、かなり薬を使っても相当難しい領域、フェンタニールを使ったとしてもなかなか痛みって止まらないものなので、かなり領域としては難しいかなという中で、なんかこういうのって神経に触っちゃってる部分があって、そういうのって結局どうしても取れないとかですね、そういうのがあると思うんですけど、ちょっとまた別の言い方すると、これ、投与経路っていうのは、全身性投与になるのか、それとも局所の
局所です。
局所の導入中みたいになると、
そうです。
そうすると、なんていうか、場所によってはやっぱ打ちづらいところってかなり多く出てくると思うんですけど、
ニードルが届かないところは、カテーテル投与。
例えば肺とかですね、脳とか、かなり場所によっては難しいと思う。その辺の注射の、なんていうか、注射のその経路、方法に対してのなんか工夫みたいなのは何かございますか。
特に今のところ医師の主義によるところが大きいと思っています。それと、すべてのがん患者さんが対象になるとは思いません。
はい、わかりました。はい、
患者さんの負担を軽減することをまずプライオリティに挙げています。
Any other questions, please? Okay, awesome. Thank you. Thank you so much. So next we have Yasuhisa Nagasaki with Neon.
Sorry, my English is not good. Incorrect word choice. Sorry. We are now Shibuya startups and microbiome startups for well-being. Neuron from neuron. Our issue is feeling trapped from pandemic, economic instability, war, and disasters. One solution, gut microbiome. Gut microbiome, brain gut axis works. Brain and gut and intestinal bacteria interaction. We regulate intestinal bacteria outside to inside intestinal bacteria character. Our product is microbiome capsules. Now I culture many bacteria and Finland microbiome pattern extract analysis of human microbiome that enable well-being transfer to product. Now prototype alpha. Market size is a very huge market. And
product positioning is at first we aim to supplement category, and we make top line, and in future we aim to medicine category. My team building now, and summary. For example, Yakult is a one strain. Our product is a some strain, a mixture, and traction and roadmap. Neuron incorporation in 2022. Go to market 27 and expand to global 28 and 30 in the IPO. Thank you.
Thank you. Questions? Any questions from the judges?
Okay, I have. So the Asian.
できればすみません。クエスチョン日本語でお願いしていいですか。
ご存知かと思うんですけど、日本人とかアジア人とコケージャン、その白人のマイクロバイオームのコンポジションがすごい違うっていうのがあると思うんですけど、製剤的にはこれはその地域ごとを目指すのか。
いや、まずは自分でセレクトしてきた今着目しているのはセロトニンであったりオキシトシンだったりとかドパミンだったりとかいろいろホルモンあるんですけど、それに着目してまずは順番を決めようと。菌の順番を決めて、その中で、可能性があるところの組み合わせっていうのを見つけてきて、っていうところからですね、最初は本当に決まりきったプロダクトからどんどん出していくっていうことを考えてます。
あと今後のその実験のモデルの問題なんですけど、やっぱりこのマウスのマイクロバイオームと人のマイクロバイオームが完全に違うっていうことなので、マウス使えないっていう開発計画になると思うんですけど、どういうもので PD とかを取ってですね、
今一応考えていたのはマウス使おうかなと思ってたんですが、当然製薬企業さんとお話をしたときに猿使わないのという話は当然ありまして、で我々ですね、今医科歯科大の設備にこの左上ですねに入らせていただいて使わせていただいているので、そのあたりでちょっと猿のモデルっていうのも検討するかなというふうには思ってます。
猿を使ってくると、またその病態のモデルっていうのがまず使いづらくなってしまうと思うんですけど、そこの病態でなおかつ人のマイクロバイオームを再現したっていうものに関してはどういった実験系を計画されていますか。
それはすみません、まだちょっと計画自体が決まってない状況で、これから検討していくところですね。
Okay, thank you. Any other questions, comments? Okay, thank you so much. So next we have Yoji Otsuki with Cubic Stars.
Hi, hello everyone. My name is Yoji from Cubic Stars. So Cubic Stars is a company originated from University of Tokyo and the Riken Institute. So we provide the service of the 3D imaging of the organ and the data analysis service for the pharmaceutical company, especially in the preclinical stage, by using tissue clearing technology called Cubic. So our customer are the scientists, especially in the R&D division of the pharmaceutical company. So here's what they do. They have to spend a lot of time, I think most of you are doing right now, cutting the, inject the drug candidate and make into the slicing every day and take the samples. And in the end, to see the situation in the 2D. So it's a very time and energy consuming process, but it's very hard to see the whole picture of the organ. And also very difficult to get the right information they need. So here is what we do. So we provide the whole organ 3D imaging view with the cellular level. So here's an example of the mice brain. So we will let them see where the drugs are going, what's happening inside the body in a very detailed level, at the cellular level. So basically, we're going to updating the current PK or tox testing from the current 2D level into the 3D level in order to accelerate the drug discovery timeline. So here's the traction. We actually started our service this February, and then so far, 7 out of 10 Japanese pharmaceutical companies are our clients. And so far, we are getting positive testimonials from the clients, especially for the clients who are doing the research in the CNS region. So here's our team. So we are made up of the team of the top scientists from the University of Tokyo and the business leaders who has a global experience. I'm from the business side. And since we are generating a revenue a little bit this time, so we are not preparing the fundraising yet, but I would love to get the opportunity for the mentoring and the feedback in order to expand our business globally. Thank you very much for your listening.
I ask a question about parameters that you can measure with your technology. You showed us a very high resolution image of an animal brain, and I wanted to understand what kind of parameters you can measure with this technology.
All right, so we have more than 20 types of the proteins. So depending on what they want to see, you can see on the this target. This is a small example, but if you want to see the neural activity, we can stain the CFOS. Or if you want to see the blood vessel, we do the immunostaining basically. So if in that case, we use the CD31. So depending on the customer wants to see, we can conduct the 3D immunostaining. So that's the technological background of this technology.
So, many neurodegenerative disease patients are suffering from some accumulation of toxic proteins inside brain. And can you visualize those toxic protein accumulation in animal model?
As long as we can stain in the 3D 3D immunostaining, we can see the statistical data. So the key point is not the imaging. So we provide the data sets and data analysis, and depending on what the customer wants to see.
Are you currently be able to measure TDP-43 or beta amyloids or tau proteins? These are measurable right now?
Not everything, but we have various kind of protocol. So in case of the new target, we conduct the co-studying, contracting with the pharmas. So we are supporting them to visualize those difficult targets.
Thank you. Very impressive. Thank you.
I know you're starting in the brain. Can this technology also be applied in other organs as well?
Yes, I emphasize the brains, but basically any types of the organ can be the targets. And recently, for instance, spheroid or organoids, many clients want to reduce the animal testing. So we can support the service for the organoids testing as well.
Since you have so much data, could you apply somehow machine learning to do even predictions, or are you planning anything like that?
Not yet. So we just started our service, but we would love to get the opinion from your side. But we do use the AI modeling in order to, we have the software.
Yes.
In order to get the answer as soon as possible.
Okay. Thank you.
Thank you.
Thank you so much.
Thank you so much.
So next we have Yuji Otsuki. Oh, no.
So very, yeah. Sorry, very similar name. My name is Yuji Otsuki. He is Yoji Otsuki. Okay. Sorry. Thank you. Okay, again, my name is Yuji, CEO and founder of Ferrotcure. We are developing ferroptosis-inducing anti-cancer therapy. 90% of relapse and mortality is caused by resistance to anti-cancer therapy. I was a surgeon. I have experienced many of those patients, and I also lost my best friends by relapse of cancer. So we'd like to save them. Oxidative stress is cell waste causing damage to the cells, and ferroptosis is cell death induced by oxidative stress. But cancer cells can survive although they are strongly exposed to oxidative stress. The reason that they have mutated this defensive antioxidant system to avoid oxidative stress. This adaptation is very critical for their survival because antioxidant system can cause drug resistance. So we induce cell death by weakening this antioxidant system in cancer cells. Our drug show high effect. We could reduce tumor volume by 83% in colon cancer mouse model, and this result is reproduced even in other solid cancers, breast cancer, lung cancer, and so on. And there are only few side effects. There is no weight loss, very safety. We are planning to start phase 1 clinical study in Japan in January 2024. And we intended to raise $20 million January 2025 for phase clinical study in US. There are 3 companies which are developing ferroptosis-inducing anti-cancer therapy. But our drug show higher effect while being safer. And we anticipate to enter the clinical situation first. We expand our indication from triple-negative breast cancer to all breast cancer and all epithelial cancers. Please join us. We are looking for collaborators, investors, and some experts. Thank you for listening.
Thank you.
Thank you so much for your presentation and very interesting. And several questions actually. So antioxidant approach is not so cutting edge of science. There are so many long history and almost the drug is failed. So I'd like to know what's the difference of that. So ferroptosis is one and only the way to solve such an issue. That is question 1. And question 2 is about I suppose you are targeting is XCT plus LDH. So I suppose that is also the old target, and I'd like to know about what kind of IP strategy and also entry barrier you would like to secure.
Yeah, thank you. One answer for one question is that ferroptosis, old technology, but our target XCT and LDH is our first technology, and in the past, ferroptosis is the ferroptosis-inducing anti-therapy use only one target. This caused the resistance to ferroptosis-inducing anti-cancer therapy, but we use 2 targets. This 2 target is used by cancer cells to avoid oxidative stress strongly. So we induce strong cancer cell death by using 2 targets. That is one question. And sorry, I forgot. Thank you. Yeah, IP strategy. Thank you. Yeah, as you know, we also we use 2 old drugs for XCT and LDH. So and like Felix, we have the application patent and formulation patent and selection marker patent. We will apply those patents so we can break the barrier. You mean why we choose triple negative breast cancer?
So the patient stratification strategy, inclusion, exclusion.
Yeah,
something like
thank you. Yeah, in triple negative breast cancer, our target, XCT and LDH is promising target, is noted as promising target. And we have already get some candidate for selection marker, and we check whether those candidates can work as a selection biomarker in phase 1 clinical study. And from phase 2 clinical study in US, we choose those selection marker to select recruiting patient as criteria.
I kind of had the same question, but so is TNBC a ferroptosis sensitive tumor type? And if it is, what assay do you use to determine ferroptosis sensitivity?
Sorry?
How do you determine ferroptosis sensitivity? I assume you chose TNBC because it's kind of ferroptosis sensitive, correct?
Yes, that's right.
Yeah.
Yeah. For example, TNBC or renal cancers are the reported that ferroptosis is effective cancer.
But second part of my question was, what assays do you use or what kind of analysis do you use to determine whether a tumor type is ferroptosis sensitive or not? To choose your indication strategy.
Sorry,
ですね、感受性、どのがんがフェルトブスが効くのかっていうのを
Yes, yes,
デターメンするためにどのようなアッセイを使用されているのかっていうちょっと興味があったんですけど。
Yeah, I know your question, but so it's a very confidential method, so I can show. Sorry, I can show. Yeah, later I explain you.
Yeah, thank you so much.
Yeah, thank you.
So next we have Nicholas Muliadi. Nicholas is with Tupac Bio.
Okay, lucky last, let's get this out of the way. Hi, I'm Nick Muliadi. I'm the CFO of Genominer. I know we registered initially as Tupac Bio, but we rebranded to Genominer about 2020. We do bioinformatics and genome mining, which is basically asset targeting with artificial intelligence and machine learning. We provide software platform solutions for biotech companies and license assets from our proprietary microbe library. And we also use our platform internally to sequence our bacteria and predict what compound they produce for commercialization into biopesticides, pharmaceutical, or even other probiotic ingredients. And we also use it to produce high quality gene data that we can use for other applications as well. We are currently post-revenue with an ongoing feasibility study series with a Japanese chemical company. And we've had inbound M&A interest for our bioinformatics software asset. As I mentioned previously, we are a Delaware C Corp with a As I mentioned previously, we are a Delaware C Corp with a Japanese kabushiki kaisha. We've been active since 2016, but we pivoted into our current bioinformatics focus starting in 2020. And the founders were PhD students in Todai, and I was the first non-founder hire, and now I'm the CFO of the company. That's it for me. Thank you.
Thank you. Any questions, please?
Yes. Could you tell us a little bit more about how your algorithm works? So like what kind of data inputs you collect and then put into your system, and then what exactly does it produce? How do you use it? At least you can explain it in simpler terms, right? So that's perfect.
So basically what we do after we have the clean sequence data of the bacteria in question, we can run it through our pipelines to analyze for specific byproducts that they make. And even if we have novel compounds that are possibly present in the bacteria sequence, we can use our predictive algorithm and machine learning to predict what are the most possible similar compounds that are already cataloged in it.
Okay, so you find the compound, and then the functionality of the compound is determined by the similarities to where you find the.
Yes, it can either be the similarities or the existing compounds,
Okay.
or even for like the very novel compounds, then that requires additional work to sequence or like to figure out
I see.
what the possible applications are.
It's a bit like a natural product kind of
Yes.
screening, and then you find the compound.
yeah.
Okay, interesting. Okay.
Yes.
Thanks.
Please correct me if I'm wrong, but I suppose Ginkgo do the similar things, but could you please let me know the differentiation?
I'm not familiar with what Ginkgo's been doing right now. We've been aware of them for a while, but I can't answer that question right now. Sorry.
Okay, awesome. Yeah, thank you so much. Thank you. So we have one or two more additions for the judges. Please add Koshin. Is Koshin still here? With Provigate. It's not on your list, but please add the name. Oh, you're going to use yours?
Just to show my company's name.
Okay.
Okay, I've just decided to join in today's presentation just a while before, so I don't have any presentation deck, so I'm going to present by myself. So my name is Koshin from Provigate, and we are the glucose monitoring device company. And when we talk about the diabetes, people often say that it is quite difficult to change the behavior of people with diabetes because they love to eat, they cannot stop eating, they are stubborn and they are lazy, and it is quite difficult to change the behavior of those kind of people. But this is wrong. It is consensus that people with diabetes, many studies have proved that people with diabetes do change their behavior even without any intensive education, if provided the enough glucose monitoring device for their home use. So, but the problem is today's glucose monitoring devices need needles, right? So it is painful and expensive, and sometimes it is overwhelming. So that's why more than 90% of people with diabetes do not check their glucose levels properly in their home. So that's why we focused on the different way. So we focus on, I have some, the model. This is albumin molecule. We focused on glycated albumin. So glycated albumin is an indirect biomarker to monitor the glucose levels. So you can count how many glucose molecule on the surface of this protein. So this process is, called glycation, and this is proportional to the glycate level of the last 1 week. So you can count how many glucose molecule on it, and then you can get easily the week average glucose levels. So this is how to use this molecule. And the good thing about glycated albumin is you can check it through your saliva sample very precisely, very accurately, without any pain. So that's why we decided to utilize this biomarker to make the home glucose monitoring device. So we have developed the home glycated albumin monitoring system, and then we have done the phase 1 and phase 2 clinical trial in Japan. And the result was astonishing. So more than 90% of participants have improved their behavior and then improved their glycemic control. So it was surprising. So this is the world first, completely non-invasive and the behavior changing methods for the people with diabetes. So we are now planning to expand our activities in the United States, and we are planning to do the phase 2 and 3 clinical trial, hopefully in the 2024. So that's why I'm here to, so why not join this incredible journey? So please get in touch with my company. So thank you so much. Bye.
Thank you. Any questions for Koshin and Provigate?
Yes.
No, I'm asking if the judges have any questions. Okay, we have one. Yes, please.
Thank you for your presentation. Glycated,
albumin.
Glycated
Glycated
albumin is your
yes.
product, right?
Yes.
And currently, glycated hemoglobin is used as a standard,
Okay, yes.
long-term glucose level monitor, right?
Yes.
And do you have goal to replace, existing, long-term glucose exposure?
Okay,
marker?
not really. Glycated protein is like glycated hemoglobin or HbA1c is gold standard for the diabetes diagnostics. So, but the difference between the glycated albumin and glycated hemoglobin, it's half-life. The glycated hemoglobin's half-life is more than 117 days. That's why it doesn't change for a long time. So even if you change your behavior for 1 week or 2 or 3 days, it doesn't change. It doesn't change at all. So basically, if you monitor the hemoglobin, so you will be disappointed about, I tried so much for the last 1 week, but my A1C doesn't change it at all. So people with diabetes will, demotivated. But when it comes to glycated albumin, if you change your behavior for, let's say, 2 or 3 days, and then it goes down. So it's very responsive. So that's why. So we are not thinking about replacing the hemoglobin, but we are thinking about replacing the SMBG or existing glucose monitoring device. But it's more than glucose monitoring device because we also provide a smartphone application as well. So we visualize the changes of glycated albumin level. So it combines with the application and the device. And then as a whole, it works as a digital therapeutics. So it's not replacing just the glucose monitoring. Yeah.
Understand. Thank you.
Thank you.
Yeah, and I want to understand, how the short-term glucose changes can be monitored by your customer. Could you describe, type of device they have to use?
Yes. What we are trying to make is this sized biosensor, and this technology is using the enzymatic reaction. And this method is very similar to the glucose monitoring sensors, but we monitor the glycated albumin instead of glucose. So the enzyme is very different from the glucose sensors, but basically the same, using the same technology. Yes.
They put his or her saliva, into.
Honestly speaking, saliva is. Final, how should I say? In the final goal is, of course, to make the device for saliva. But at this moment, we do not have enough sensitivity for saliva samples. So for the saliva for this biosensor, you can use blood samples, but it's only once a week. But when it comes to saliva samples, we can do use HPLC. So at this moment, we have just launched our service in Japan, and then the users will send their saliva, and then it will post in, and then send to our laboratory, and then we can monitor the glycated albumin within a day or two, and then back the results to their smartphone. So this is how does it work.
Thank you.
Yeah. Thank you so much.
Thanks so much.
So that concludes the pitching activity for this workshop. So let's give it up to all of the entrepreneurs. Thank you so much. So now we're going to take about 5 minutes break while we speak to the judges to tally up to see the top 5 that they've selected. So give us about 5 minutes.